everyone, welcome to our Glory Globe TV's really breaking news. Scientists have engineered the first fully human-made bone marrow systems, recreating the complex mix of cells, vessels, and nerves that normally live deep within our bones. The breakthrough lets researchers study how blood is formed and how it fails in disease like cancer without depending so heavily on animal models by building a realistic blood factory in the lab. The team opens the door to safer drug testing and eventually personalized treatments built from a patient's own cells. Yeah, the bone marrow usually does its work without drawing much attention. It becomes noticeable only when something disrupts it, such as blood cancer. When that happens, it becomes essential to understand how blood is normally produced in the body and what causes this process to break down. For many years, scientists have depended on animal studies and simplified lab grown cell systems to investigate how bone marrow functions. Researchers from the Department of Biomedicine at the University of Basel and Rudy Hospital Basel have now created a detailed bone marrow model made entirely from human cells. According to the team led by Professor Ivan Martin and Dr. Andres Garcia, these new systems could support research on blood cancer, provide a platform for testing medications, and possibly contribute to personalized treatment approaches. Their findings were published 18 November in Cell Stem Cell. Viewers, bone marrow contains a variety of small specialized environments known as niches. One of the most important niches for producing blood cells lines near the bone surfaces and plays a role in why some blood cancers resist treatment. And this area called the endosteal niche contains blood vessels, bone cells, nerves, and immune cells. A human bone marrow model that includes all of these components had not existed until now. The research team has now assembled a model that incorporates these features. They begin with a synthetic bone scaffold made of hydroxyapatite, a substance naturally found in human bones and teeth. Human cells were reprogrammed into pluripotent stem cells using molecular biology techniques, enabling them to develop into many different cell types in response to signal. These stem cells were then placed into the artificial bone scaffold and guided through specific developmental stages to generate a diverse set of bone marrow cells in a consistent, controlled manner. So analysis showed that the resulting three-dimensional structures closely matches the human endosteal niche, and it's larger than earlier attempts. With a diameter of 8 millimeters and a thickness of 4 millimeters, with this model, the researchers were able to maintain human blood cell production in the laboratory for several weeks. Amazing, sir. May this exclusive report, Glory Globe to be Delhi, Bharat.